This video is going to walk you through solving genetics problems that involve epistasis. Epistasis occurs when a gene at one locus, and that word means just location in the genome, alters the expression of another gene at another locus. So in this example, we're looking at two genes that control the coat color in mice. And one gene you can see is going to be denoted with B's that codes for either black or brown pigment. And you can see over here in our key that it's going to be black if they have at least one dominant allele. It's going to be brown if they have two recessive alleles. The other gene is denoted with the letter C, and that gene is going to determine if the pigment gets deposited. So an animal would be white if they don't have any dominant version of that allele. So in this case, we have three possible phenotypes. These are the parents. They are heterozygous for these two traits. So essentially, this is a dihybrid cross, just like the one that we did before, looking at peas. But in this case, the interpretation of the phenotypes is going to be different. So we are going to set this up just like we did the other dihybrid cross. And we are going to assume that each of these parents can make four different gametes or have four different combinations of these two alleles in their gametes. So I'm just going to go ahead and write the gametes down both axes of this square. So they could have big B and big C in their gametes. They could have little b and big C in their gametes. They could have a big B and a little c in their gametes, or they could have a little b and a little c. So what's true of one of these parents is going to be true of the other because they are genetically identical in reference to these two traits that we're considering in this epistasis problem. So I'm going to write the same gametes across the other axis of the square. Okay, so just like we did with with all the Punnett squares, the next step is to populate these squares. And what we're doing is we're imagining the fusion of this gamete with this gamete with this gamete. So we're populating the squares with the diploid genotype of the offspring resulting from that fertilization event. So I want you to pause the video and fill in the rest of this Punnett square by taking a look at the genes in, or the alleles in this gamete, and then populating the square, adding them together basically for each of the squares. Look at the columns, look at the rows, and populate the square. So you should have come up with these genotypes in your square, representing all the possibilities of combinations of alleles in the offspring of these two parents. So now you're going to do what you've done for all of the genetics problems. You're going to look at the genotypes that you've written, and you're going to determine what the phenotypes are based on your phenotype map. So remember, if the animal had an uppercase B and at least one uppercase C, they were going to be black. So I see 1, 2, 3, 4, 5, 6, 7, 8, 9 animals that are black. Remember from our key that if the animal was homozygous recessive for color and had a big C, meaning the pigment would be deposited, then they were going to be brown. So I see 1, 2, 3 animals that are brown. And then if the animal had 2 little C's, that meant that it didn't matter what was going on with the bees. No pigment would be deposited. So what we see here is 4 animals having 2 little C's, meaning 4 white animals. So this is a 9:3 to 4 ratio. With a typical dihybrid cross, we would expect a 9:3 to 3:1 ratio, and often we see variation from that ratio with epistasis.